Hey, welcome to the second attempt at getting this voice recording for you so that you can review it from home and be prepared for your second exam on Biochem. So the first image here is showing you water molecules. So make sure that you understand the makeup of a water molecule, oxygen being the center and the two hydrogen being the ears. The reason that it's bent, we have a covalent bond between the hydrogen and the oxygen, but unlike a regular standard covalent bond, this is a polar covalent bond. The reason it's polar is because the oxygen tends to be more electronegative, so it tends to pull the electrons more towards it. Since it's pulling more electrons towards it, electrons having a negative charge will make the oxygen atom more negative, making the hydrogens more positive. The polarity is going to play out a big part, especially when we're talking about hydrogen bonds. From here on out, you need to understand that any water properties we talk about are due to these hydrogen bonds. So this image is showing you how density plays out, the uniqueness of density with water, and that if we increase density going from left to right, it's actually the most dense stuff is the liquid, then the solid, then the gas. Looking at the images, you can see the hydrogen bonds become much more stable at a lower energy level, right? So we have lower energy here, and this has a higher energy. And the more energy you have, the faster the hydrogen bonds get broken, hence the water molecules can be a little bit closer together. In the lower energy states, the hydrogen bonds become more stable and tend to be spread out. And this is going to be vital for life on this planet because ice then floats on top of its liquid water counterpart. Next image, water strider. If you remember seeing images of this or videos of this. Also the Jesus lizard would go with this, or the basilisk. Water strider, you can see, is depressing that water surface yet not breaking it. So we're going to call this surface tension. And why is it that surface tension exists for water? Hydrogen bonds, just like I said. So hydrogen bonds are going to be the reason why. So it's resisting the surface breakage because water tends to be cohesive, and that's water sticking to water, right? Whereas adhesive would be water to other things. So here you're seeing an example of adhesion and cohesion in play. So which part of the picture would be cohesive? The water droplet. The reason water makes droplet is because water will stick to itself. The adhesive part is the actual component that the water is sticking to the leaf. This image, not Darwin, not Santa Claus or Tinta Claus or any other donation of what this bearded gentleman is. This is showing you, I don't know if you can see it, a water level in the balloon. All right, so the reason he can hold it under the flame would normally warm up the material that's on the bottom side of the balloon, and if that balloon was empty, no water in it, that polymer would stretch out and the air would force itself through the weak spot, popping the balloon. But instead we're filled with water, and water is continually taking in that heat, but the temperature really isn't increasing because water has a 4.18 joules per gram degrees Celsius, or 1 calorie per gram degrees Celsius specific heat. Remember, specific heat is the amount of energy it takes to heat 1 gram of a substance 1 degree Celsius. And water's is incredibly high. So that water in that balloon is taking in that heat, yet the water isn't really changing temperature. Hence, the polymer that's laying against that water isn't changing temperature either. So we got a nice buffer. Understand that the specific heat of water allows for environmental conditions that you see, like lake effect near Lake Michigan, or the absence of the water that you see in deserts with the variable fluctuations. I think that's repetitively redundant, but the fluctuations that you see in temperatures from morning to evening. Methane man. Methane, right? CH4. Why is it so important, or why is it a Well, this is a base model, or a basic model, of an organic compound. Remember, organic means that it contains carbon. Methane is one of the byproducts of protein breakdown, and hence you end up getting it in gaseous productions. This is methane. What you need to understand about methane, or carbon more specifically, is carbon is tetravalent. Tetra meaning 4, valent meaning valence electrons. Wow, can spell on the smartboard? Yes, I can. All right, so valence electrons. So the interior ones are not valence. Valence means outer shell. So the farthest out shell for carbon are these out here. Right? And these are the ones, valence electrons are the ones that are most important in participating in reactions. So you can see carbon here is sharing electrons with hydrogen. So hydrogen's electrons are the X's, so that hydrogen has 1, that hydrogen has 1, this hydrogen has 1, and this hydrogen has 1. And the sharing component, right, covalent bond, is showing you that carbon has 4 bonds generally to fill its octet rule. So on the outer circle, carbon now has 8, where it originally had 4. So this is a very stable carbon compound now. And hydrogen is happy as well. Next image. We have R, meaning rest of the molecule, meaning we could tag and add however we want here. But the big important part is this OH, right? So we're talking about a functional group. Hopefully you already know. This is hydroxyl. R on both sides, meaning we're building the molecule from the right to the left, right and left, I should say. So it might look something like this. Right, so we have rest of the molecule coming this way. We have rest of the molecule going this way. So we're looking at this C double bond O, this corner, anytime you see a corner in organic chemistry, so it might be even written like this. All those corners represent carbon. So we're seeing C double bond O. You should know this is a carbonyl. And since it's in the middle of a molecule, it is a ketone, carbonyl. This would be an example of a aldehyde carbonyl, because we have rest of the molecule here, but there's an H. Why an H? Because it's the end of a molecule. So it would look more like this. Right? Remember, carbon has 4 bonds. You can see 1, 2, 3, 4 around it. C double bond O is the important part of this. Right? So that is the carbonyl. And since it's off the end, it's an aldehyde. Please don't judge me on my spelling. Last piece here, we've got rest of the molecule here. So the most important piece is this, right? It looks like a carbonyl, but a hydroxyl at the same time. So this is carboxyl, you guessed right. Nice job. Bonus point for you. And it's acid-based, right? The reason it's acid is because this H, when you put it in liquid, tends to dissociate H pluses. Remember that concentration of H pluses govern pH. So if we increase the concentration of the H pluses, our pH goes down. I know that's kind of hard to understand, but if you've taken some chemistry, the negative log of the concentration is actually the pH. So it ends up coming out that something like 3.0 times 10 to the negative 4th concentration of H pluses actually has a pH of 4, or around there. Next molecule, rest of the molecule. So we're looking at the NH component. This is amine. So between the last one and this one, we would have an amino acid if it was on one side and the other. Rest of the molecule, SH, sulfhydryl. Oh. PO4, right? You got the phosphate in the center, so this is a phosphate. Where do we see phosphates? Hopefully you responded. Nucleotides, right? Because you have sugar, phosphate, nitrogen base. And you also see it where? Phospholipids. Nice job. Positively north ticket for you. I'll give it to you on Saturday, because today's Friday. Where would you see this one? Disulfide bridges. Reviewing, this is going to be a main component in the tertiary structure of protein folding. Right, so keep that in mind. All right, on to the biomolecules. So what we see here is they're all sugars. How do we know they're sugars? Remember, you're looking for that CH2O combination. I know if you decide to sit here and you pause this and you count it all up, you look at the disaccharide, you would say, oh, disaccharide, right? That would be 2 monosaccharides. Well, if monosaccharide is C6H12O6, all I have to do is multiply it by 2 to get a disaccharide, right? 2 of them being put together. And you would be correct, but it doesn't work out to be exactly this. Why? Well, you know that when you put 2 monosaccharides together, you have to pull out water. So if you were to do sucrose's formula, it would be C12, H, you would think 24, but it's actually 22, O11, because we're taking out an H2O. So if we added those in, then it would be 24 and 12, right? So keep that in mind. So disaccharides, polysaccharides, right, are all put together by dehydration reactions, right, or condensation reactions. So here, right, they're all going the same way, right? Here we have the same thing. We're seeing glycosidic linkages, right? You need to make sure you know the name of the bonds of these macromolecules. You need to know what the monomer name is. You need to know what the polymer name is, right? And since these are going the same way, this is a hydrolyzable by animals type of glycosidic linkage. It's an alpha glycosidic linkage. Next image is showing you another polysaccharide. I don't know if you've identified it or if you can read it, but it says up here, cellulose, found in plants, right? So you can see the glycosidic linkages. That one goes down, this one goes up. It's a beta glycosidic linkage, which means it's non-hydrolyzable by animals, so we can't break it down. We need some other symbiotic relationship in order for this to happen. So that symbiote is usually bacteria or protists if the animal is eating cellulose high in their diet and not just using it as a source of fiber. They're actually using it as an energy source like cows, goats, rabbits, and so forth. Next image is showing you hydrolyzable again. This would be glycogen based on the branching that we're seeing here. So all these subunits within, right, all these glycosidic linkages are all going the same way, right? So our body has the enzymes to be able to break these down. Next image. Remember I said that this accordion-like shape is actually showing you C's at each one of these junctions, right? When you get in organic chemistry, you have to assume, if you see nothing else, that, for example, this carbon on the end here would have 4 bonds, so it has 1 bond to this carbon, and then it has an H here, an H here, and an H here. So that carbon has as much hydrogen as it can. This one would have hydrogen, hydrogen. So what you're seeing is long tails of carbon and hydrogen. That's why they're fatty acids. Where does the acid component come from? The C double bond OH that was here, right? And the reason it's not OH now is because water was removed to stick it to the glycerol backbone. Right, that removal of water allowed for this linkage, and it's called an ester linkage. What are triglycerides? That's what this is. Mainly used for energy stores, specifically in your love handles, right, or your spare tire, or in the case of my father, the full-blown keg he has around his waist. Phospholipids. These are another grouping of the lipid category, very unique in that they have a phosphate head, they have the fatty acid tails, right? This one is actually showing you an unsaturated fatty acid tail because there's a double bond, hence why it's kinked. But the unique part of it is that it has a polar region and it has a non-polar region. Why is this important? Well, phospholipids are mainly used in cell membranes. Keep this in mind because next unit we're going to play this out in much more detail, right? And you'll see phospholipids more represented like this, where we have the tails facing each other and the heads away from each other, because outside the cell there'd be lots of water, so we're polar environment with the water, and then inside your cells is very water-based. You're going to see polar with the water environment, and all the nonpolars in the center oriented towards each other. So since this molecule is polar and nonpolar, the vocab term that goes with it is amphipathic. Okay, so make sure that you keep that in mind. That's a big term. It keeps coming up throughout the year. Next class of lipids. This one is cholesterol, as you can see here. Right, it's used in two different ways. One is for sex hormones, right? So we're using this to make testosterone and estrogen. It's also used as a stability molecule in cell membranes. So I know what you're asking yourself. All these molecules I have to be able to identify just by looking at them? And the answer is yes. So you got to start picking up on how these molecules look different yet look similar. So you've seen rings before, right? The difference is here that these rings are fused together, and there's 4 of them that are fused together. That's a key component for cholesterol. So as opposed to when we saw a sugar molecule had spacing between it, right? So those rings aren't fused together. And if you see a nitrogen base, yes, there will be 2 rings, and it's possible that they're fused together, but we don't have 4 of them, right? And if it's a nitrogen base, there's going to be a heck of a lot of nitrogen in those rings, right? So keep that in mind. We don't see any nitrogens here, and we see 4 rings. So this is cholesterol. It's used for sex hormones and cell membrane stability. All right. Next component, you can see it says here on the slide that we're talking about peptides, right? So we've got carboxylic acid, we've got the amine. So this is showing you 2 amino acids coming together by pulling out, right, H2O, and together we make a peptide bond. All right, so water is being pulled out so that we can link these 2 together. So the monomer is an amino acid. We're forming a polymer, starting to, via a peptide bond. So here you can see we've now put a whole bunch of amino acids together in a chain, right? And that chain of amino acids and their order is the primary structure of protein conformation, how it folds together. This image is showing you how one amino acid farther up that chain that we just showed you is interacting with an amino acid farther down the chain, right, through a hydrogen bond. So this is an alpha helix. Do not confuse it with DNA that is a double helix, or RNA that has helical structures. We end up seeing this when we're folding up a protein in a local arrangement, so in a much more closer, not bigger picture type of arrangement. But all these conformations, all these folding components, right, so this is secondary, all these folding components boil down to how the enzyme actually works, form fits function. The other one that you need to keep in mind is there's beta pleated sheets as well, right? And that's when the chain kind of does this thing. That was horrible, right? Hydrogen bonds kind of come across. See, I got an extra point on that one. But we end up getting this folding of the amino acid chain as opposed to a helical structure. Keep that in mind. Last piece here, a big part of this that you need to make sure you're aware of is this disulfide bridge. It's a strong covalent bond, right? That's going to add to the stability of this molecule overall. So this is tertiary structure, right? This is going to be found in every single protein that you find. Every single protein you find at least goes up to tertiary structure. Some of them will go up to quaternary structure, but that's only if the protein is a little bigger in size. So something like insulin will only have up to tertiary structure. Something like hemoglobin here has quaternary structure because you're seeing 1, 2, 3, 4 of those polypeptides that have all the way up to tertiary structure, all interacting together to make this one big protein that has function. So if you remove one of these polypeptides, the protein doesn't work anymore. Or if you have an alteration in one of these polypeptides, it might not work anymore. Because remember, form fits function. Right? So you're seeing hemoglobin in this image, and this is the quaternary structure of protein folding. Finally, moving into nucleic acids, this is what your monomer looks like, right? And this is called a nucleotide. Right, so nucleotide has a nitrogen base, a sugar, and a phosphate. Remember that nucleotides can build DNA, and they can build RNA, right? But we also saw this in an image where we added 2 extra, I should say, on this image, phosphates. So we end up having a molecule called ATP, right? Or we cleave off one of these phosphates, and you end up having a DP. Regardless, these are energy sources for organisms too. So not only is it a genetic material, but it also can be an energy source within organisms. This we know is a nitrogen base that only has 1 ring, right? But you see the nitrogens, that's why you know it's a nitrogen base, right? So 1-ring ones are C, U, and T, right? Cut by the pyramid, right? Or King Tut lives in a pyramid. And I'm sure I spelled that wrong. Pyramidine, I think, I-N-E, right? So, or if you step on a teeny tiny mouse pyramid, right, that would cut you. Whatever it takes for you to remember, right? And if you have 2 rings together, right, with lots of nitrogen in it, this would be a nitrogen base as well, but this would be a purine, right? Something uncut is very pure, and that would be like pure gold or pure silver, right? A's and G's. All right, so this is showing you one of those purines where you end up having 2 rings together. Last image is DNA in its bigger picture here. So you can see we've got sugars and phosphates going down the side, right? So this part of the DNA molecule is all going to be sugar-phosphate bonds, and that's a phosphodiester bond, right? Which is a covalent bond, right? So it's strong. Whereas across the center, you can see we end up having hydrogen bonds, which are weaker. You need to understand that that's important because if we can't open up the DNA molecule, breaking these hydrogen bonds, we can't get to the code, right? And the code are these nitrogen bases that you find on the inside. G to C has a 3 bonds, so we know that C is going to be this one, and G is going to be this one, whereas in this one, the one below it, I'm sorry, I'm saying this one, and you can't see where I'm pointing, the one that's below it with 2 bonds is going to be A and T, so T would be this one because it's smaller, it's a pyrimidine, and A would be this one. Not that you have to memorize that on the test, but if you can put all those pieces together, that'll help tremendously in your understanding of how all this works. And this concludes the first review session for water, carbon, and macromolecules. If you have any questions, email myself or your teacher if you have Mr. Murphy, and we'll get back to you as soon as we can. Look for the next review session to be on enzymes. I wish you the best of luck in your studies, and we'll see you in class.